Hi everyone, my name is Kaylee and I'm a final year PhD student in the Ocular Pharmacology and Genetics Research Group in University College Dublin under the supervision of Professor Brendan Kennedy. So I have been working on uveal melanoma research for the last 3 years since the beginning of my PhD, and I am delighted to have been asked by Ocular Melanoma Ireland and the organisers to talk today a little bit about our uveal melanoma research team in UCD and some of the research projects that we currently have ongoing. So as I mentioned, I work in the Ocular Pharmacology and Genetics Research Group in UCD, and this group was established in 2003 by Professor Brendan Kennedy, who you can see on the screen here. And while we have expanded our work to include uveal melanoma research over the last 3 years, this was previously not the research focus of our lab. So our main expertise is in ocular disease, but prior to our uveal melanoma research, we would have focused on inherited retinal degenerations and in other ocular disorders that involve abnormal blood vessel growth. So we currently have 9 different researchers in our lab that are working on a variety of different projects. But our first uveal melanoma project started with just me and Brendan back in 2017. So since then, we have been extremely fortunate to be able to add new researchers to our uveal melanoma research team. So Kaylin, Marcia and Husni, who you can see here, all started working on uveal melanoma research in our lab just this year. So Kaylin is a research assistant that helps with my project and with everyday tasks in the lab, and I'll explain my project in a little bit more detail today. Marcia is a first year PhD student who is working on the role of cannabinoid receptors in uveal melanoma, and Husni is a postdoctoral research fellow investigating the therapeutic potential of HDAC inhibitors in uveal melanoma. So as I mentioned, our first uveal melanoma project began back in 2017. But prior to this, I, like many others, had never heard of uveal melanoma before. And I don't want to dwell on the numbers here, as I'm aware that Valerie O'Neill and Noel Horgan, both of whom we work very closely with and who are the experts in uveal melanoma in Ireland, will be speaking, and no doubt they'll be talking about this paper that they published in 2018 about uveal melanoma in Ireland. But the shocking statistic for us was, and still is, that Ireland may have one of the highest incidences of uveal melanoma in the world. So for that reason, it is so important to have uveal melanoma research ongoing here in Ireland. And it's so critical that we work closely with the clinical team and with patients to achieve this. And this is something that we in our lab in UCD have sought to incorporate into our research from the beginning, and is something that we hope to continue to be able to do going forward. So before 2017, there was nobody in UCD that was directly focusing on uveal melanoma research. So how did we in our lab end up working on uveal melanoma? So although we are the Ocular Pharmacology and Genetics Research Group, we have actually done some previous research in colorectal and esophageal cancer. And over many years, our lab discovered drugs which target molecules known as cysteinoleucotriene receptors, or CysLT receptors for short. And these cysteinoleucotriene receptors are involved in inflammation in the body. So anyone with asthma may already be familiar with these CysLT receptors, as montelukast is a common drug that is prescribed for asthmatics, and this is actually a CysLT receptor blocker. So in our lab over the years, using zebrafish, mouse models and colorectal cancer patient samples, we discovered that these CysLT receptor blocker drugs, which we call Q1 and Q7, could actually prevent the growth of blood vessels, which is a hallmark of cancer, and also the growth of colorectal cancer cells. So going forward, we used our 2 lead CysLT blocker drugs, which are called Q1 and Q7. So you can see in the bottom left of the screen here. We used them in mouse models of colorectal cancer, and we found that they could actually stop the growth of tumours in these models. So that's the coloured circle that you see there, that's the tumour. So around this time, in 2016, while we were doing this research, a paper was published by a group in America which identified that actually one of these cis-LT receptors was mutated and was responsible for causing uveal melanoma in a small number of patients. So in the middle of the screen, in that blue schematic, you can see some of the most common mutations that occur in uveal melanoma. And many of you may already be familiar with mutations in GNAQ and GNA11, which you can see there, but what you'll notice is that the mutation in cysteinoleucotriene receptor 2 occurs in the same pathway just upstream from these mutations. So Brendan and I decided to investigate if we could use the same cis-LT receptor blocking drugs, Q1 and Q7, that we had found to be effective in colorectal cancer models. Could we use them for the treatment of uveal melanoma also? And that is the project that I am currently working on today. So while we may be experts in cis-LT receptors and ocular disease in our lab, we were certainly not experts in uveal melanoma when we first began this project. So throughout this project, we have been extremely lucky to work with leading uveal melanoma experts, two of which you may know and are pictured here. So Professor Sarah Coupland from the University of Liverpool and Dr. Josep Piolat from the Belvite Biomedical Research Institute in Barcelona. I was fortunate enough to win an award which enabled me to actually spend some time in Liverpool in Professor Coupland's lab. And what we did there was, using primary uveal melanoma patient samples, we looked to see if the protein levels of our cysteinoleucotriene receptors were present. So on the left, you can see an example of primary uveal melanoma tumours from patients that have low, intermediate and high levels of expression for that protein, so for the cysteinoleucotriene receptor. And what we found was that not only are the receptors found in primary uveal melanoma tumours, but that patients that have high levels of this receptor, which is shown in the red line in the graph, that these patients actually have a worse outcome than those patients who have low levels of the receptor. So that's in the blue graph. So the patients who have high expression of the cysteinoleucotriene receptor have a poorer prognosis than those who have low expression of the receptor. So for us, this was a really interesting result. And all of this data is actually from an original research article that our group published with Professor Coupland and Dr. Piolat in October of this year, along with many of our other collaborators. So that was very exciting. So following this, we sought to determine if we could actually stop the growth of uveal melanoma cells using our two drugs, Q1 and Q7, and could we see the same effect that we saw in the colorectal cancer cells. So we found that using our cis-LT blocker drugs, again Q1 and Q7, that we could actually stop the growth of both primary and metastatic uveal melanoma cell lines. So hopefully you can see in the pictures there that there are no cells growing in those that have been treated with the drugs when we compare those to the control group, which represents no treatment at all. So we wanted to see if we could have the same effect in a living organism rather than just cells in a dish. So next, we implanted these primary and metastatic uveal melanoma cell lines into the eye of zebrafish larvae, and again we treated with our drugs of interest, Q1 and Q7. And we actually saw a similar effect. So after 3 days of treatment, the number of cells, which you can see is just a little red dot on the screen there, was actually smaller compared to the DMSO control. So what that tells us is that when we treat with our drugs, we can actually shrink the number of cells in the zebrafish larvae. So that is in a living organism. So while these results were great progress and they were proof for us that our drugs could be useful in uveal melanoma, it is extremely important to ensure that we can mimic these results in a model that closely represent what's actually happening in a patient and not just in cells in a dish or in a zebrafish model. So So with this in mind, we've been working very closely with our clinical colleagues in the Royal Victoria Eye and Ear Hospital to collect patient samples, which are critical for our research. So these samples, as you know, are rare and they are extremely precious to us. So we really try to maximise the research in which they can be used. So we were granted ethical approval to collect a piece of primary tissue from consenting patients that are undergoing an enucleation procedure under the care of Mr. Noel Horgan. So we then use this tissue for research in two different ways. So one part of the tumour is sent to our collaborators in Barcelona, and it is implanted into the eye of a mouse to create a patient-specific mouse model in which we and other researchers can test their research drugs to see if they are effective in a patient-specific mouse model. So these models will benefit not only us, but the uveal melanoma research community as a whole. Secondly, a piece of the tumour tissue is cultured here in UCD by me under conditions as close to those in the human body as possible. So using a patient tumour instead of a cell line gives us a massive benefit because it allows us to maintain the cell-to-cell connections and the communications that would normally happen in the body, which doesn't happen when we're just growing cell lines in a dish. So what we do with these tumours is that they are cultured in the presence of our research drugs, so our cis-LT receptor blocking drugs Q1 and Q7, to see how they react to those drugs and if we can change cancer promoting factors in the body. So this research project is currently ongoing with our colleagues in the Royal Victoria Eye and Ear Hospital, and we've been extremely fortunate to be able to include 14 different patient samples in our research since this began in May 2019. And we've also submitted an ethics application that is currently under review to access and conduct similar research using liver metastasis biopsy samples from uveal melanoma patients from University Hospital Waterford in collaboration with Dr. Paula Calvert. So, although we are new to uveal melanoma research, I just wanted to highlight some of the progress that we've made over the last 3 years, and you can see that on the screen there, from publications to grants and presentations and national and international collaborations. And now that we have 4 researchers in our lab in UCD that are dedicated to uveal melanoma research, I think we'll be able to grow this progress exponentially, and we are definitely in this for the long haul. So we've been very eager to involve patients in our research from the very beginning, and we're very lucky to have established a great collaboration with Ocular Melanoma Ireland, with Joanne and Melody, who have acted as collaborators on a number of our research grants and have helped us to develop a website piece for Breakthrough Cancer Research, who's one of our funders, and also a uveal melanoma animation video with Icon, which is coming soon. So you'll see I've included a little quote on this screen here, and I love this quote. I think it's really applicable to lots of different areas, but research in particular, because absolutely we have a long way to go in uveal melanoma research, and I know most of you probably know that better than anyone. But I do believe that Irish uveal melanoma research, particularly here in UCD, has come a long way over the last 3 years. And especially now that we've expanded our research team, we will continue to push that to reach the ultimate goal of finding a treatment for uveal melanoma. None of the work that I have spoken about today would be possible without our funders, and even more so without our fantastic collaborators, both nationally and internationally, all of whom I've included on the screen here, both in the academic research setting and in the clinical setting. As someone who started from scratch in the uveal melanoma research field just 3 years ago, I can tell you firsthand how supportive and how helpful the uveal melanoma research community has been and still is in our research. And I believe that some of the best scientists in the world are working on this disease currently. Lastly, I would just like to thank all of the uveal melanoma patients that have agreed to take part in our research to date. In some of their darkest days, they are lighting the way for patients to come, and we truly appreciate your support in our research. Thank you all for listening, and that is my email at the top of the screen, and I'm very happy to be contacted with any questions or comments about our research. And lastly, thank you to Ocular Melanoma Ireland and to the organisers of this symposium for asking me to speak today and to share a little bit about our research. Thank you all and take care.
Hello, my name is Naomi Walsh and I am a cancer researcher in Dublin City University, and I'm delighted to talk to ocular melanoma patients and caregivers at this global symposium on my research in matching genomic and actionable targeted therapies in uveal melanoma. So just to say that my research is in the National Institute of Cellular Biotechnology, and we are a multidisciplinary centre of research that's fundamental and applied cellular biotechnology. We have a cancer research group in uveal melanoma, whereby we collaborate with many Irish hospitals. We have input from oncologists, surgical ophthalmologists, pathologists, and research scientists, and all together we work in order to bring more research for uveal melanoma. We're delighted to be part or to link in with the public patient organisation OcuMel Ireland, because it's so important for us to be able to translate our research both to patients and caregivers and the public. So a lot of my research really questioned whether or not uveal melanoma models were accurate for kind of predicting biological responses. So you can see here in the picture, this is usually how we grow our uveal melanomas in the laboratory. So they're flat in 2D and grown on in a plastic flask. So really what we know is that plastic is not a natural component of the body, and even growing the cells flat and polar and spaced out doesn't mirror how these tumours will grow in the eye. Furthermore, if we think about other models, such as mice, we find that some of these models can actually fail to recreate the complexity and specificity of tumours. For instance, in terms of patient-derived xenograft models, whereby tumour cells are placed into subcutaneously or orthotopically into a mouse, sometimes this takes a long time for the tumour to grow, and also you have a lot of infiltrating cells that belong to the host, so they are mouse cells. And what we find is that really this mirrors the genomic and the molecular characteristics of the tumour only about 80% of the time. And then if we look at other mice studies, such as mice that are genetically engineered, transgenic mice, where they have mutations maybe in driver genes of uveal melanoma, we find that in literature, they may develop the uveal melanoma, but they also develop cutaneous melanoma, which kind of indicates that these mutations working alone or together may not be able to fully recreate the uveal melanoma phenotype in this particular model. And I mean, finally, mice are not human. So really what we need to do is we need to be able to find maybe efficient and better models. So some of my research is interested in obviously targeted therapies. So looking at the clinical trials that are available for uveal melanoma. And we find that, you know, out of our research in cancer by itself, the success of these clinical trials is really undoubtedly attributed to the accuracy of modelling in the disease genetic and molecular landscape. And really that any kind of future advancements in cancer are really reliant on the development of more genetically appropriate laboratory models. So what we found when we looked at a lot of the phase 2 clinical trials that were open for uveal melanoma, particularly such targeted therapies such as VEGF inhibitors, ERK inhibitors, so signaling pathways that are involved in uveal melanoma. And we found that a lot of these were not progressed. So really, they weren't progressed either because they didn't have a good response, but when we retrospectively looked back, a lot of these targeted agents weren't even studied in uveal melanoma, or if they were, it was only in a very limited amount of cell lines. So while the current models in place, such as these cell lines and PDX mouse models, they have provided vital information in terms of gaining a greater understanding of the disease. But we have to recognize that these models are not ideal. They do not recapitulate or mirror the situation that goes on in the body. And as we know, like to date, no preclinical studies have effectively translated into the clinic for real meaning clinical responses. Therefore, better strategies are needed to improve like the translational relevance of preclinical to clinical translational research in uveal melanoma. So a lot of my research is really focused on organoids. So preclinical models that can effectively recapitulate or mirror the original cancer, they tend to provide high levels of predictivity and effective translation of our preclinical findings into the clinic. Therefore, using these preclinical model systems, ones that fully mirror the human situation, will really enhance and enable translational impact of our experimental data to inform future strategies for clinical impact. So my research is really focused on the organoids, and these are stem cell derived 3-dimensional culture structures that we generate in a lab. So they're effectively a mini tumor in a dish. So they're derived from the cancer stem cells, which allows for self-organization of the cells. They show heterogeneity of tumors, so they're more representative of the cancer from patients, and they can be developed to further show the different stages of diseases. So really what my previous research was involved in kind of showing how 3D organoids really do mirror how the tumors behave in the body. So really, in the pictures here, we have our cells that, literally just seeded into the wells and then grown over 10 days, where you can see this production of this mini organ-like system. And this is comparable to the pictures down the bottom, whereby the cells are grown flat. So there's very different morphology and even response to therapeutics as well. So utilizing my knowledge of organoids that we previously grown, we know that basically advancements in laboratory models such as these 3D organoids have the potential to create more accurate representation of how our tumors grow and how they interact with cells and tissues as well. So a lot of my research was really focused on identifying a lot of the factors, supplements, and components that are necessary to recreate this in vivo in the body structure and function in the lab. So here's some pictures of some organoids that we've been able to develop from uveal melanoma. One characteristic here that you can easily see is that they still maintain the melanin expression, which tends to be lost in a lot of the established cancer cell lines. Moreover, we also we're able to develop novel uveal melanoma primary cell lines. So although the primary cell line or cell lines kind of have limitations, they do have some use, particularly in high throughput studies. A lot of the literature has shown that development of cell lines has a very low success rate. However, we've been able to establish a really good flow through in order for us to develop primary cells and organoid cultures as well. And we know that our uveal melanoma cell lines are exactly that, because we were able to stain them with different markers that are specific for uveal melanoma. So a lot of the issues that other studies may have is that there's a lot of infiltrating non-tumor cells as well that can take over the cultures. So now that we are able to develop these unique models of uveal melanoma, what we wanted to do, and what I'm also interested in, is really understanding the genomics of this disease. So in collaboration with the Royal Victoria Eye and Ear Hospital, we conducted a genomic profiling study. So we had 10 paired samples, and just to date, we have sequenced 5 of these samples. So that's what I'm going to present today. So a lot of the sequencing involves data analysis. So we have a great bioinformatic called Charlotte, and really what she does is she processes a lot of the data that we get back from the sequencing. So it's really important that we have appropriate quality control, mapping of our reads, detections, visualization, and obviously interpretation of that data as well. So we'll just highlight some of the information that we've gotten to date. So one of the first things we looked at is copy number alterations. So these are just the structural chromosomal changes that we look at in the genome of uveal melanoma. And for this study, you can see that where you see green, everything is nice and normal. Where blue, we see a loss of at least one copy, and red shows a gain of a copy. So highlighting over here where you can see a high level of gain in chromosome 6 and also in the long arm of chromosome 8, and then a loss in certain regions such as long arm of chromosome 6, chromosome 15, and then the sex chromosomes as well. So we also look at it in a deeper level, whereby we looked at the somatic changes to the chromosome structure that results in kind of that gain or loss or sections. So if you think of your DNA as the book, the copy number alterations will show you the chapters. So it'll show you, what you're reading pretty much and where it is. Whereas the somatic variant analysis, it shows you the letters in the words. So we focus right down into the letters. So if a mistake or a typo happens in a word, sometimes that word, it makes no sense or it has a different meaning. So that's what we mean about looking right down at these individual somatic variant changes. And we can see in this patient here that there's a lot of kind of missense variation. And that does occur when there is a mistake in the DNA, in one almost letter of the DNA, that then changes the word. And this can result in obviously the cancer as well. And obviously we use this information then to be able to track whether or not these particular changes or typos that we're seeing can actually be actionable and targeted by specific therapies. So we do a really, really in-depth look right down to understand what kind of changes happens, the type of it as well, whether it's a single change or whether or not there's larger changes, whether they're deleted or inserted, etc. And then as well as that, we know that cancer develops in a cascade, with multiple networks working together. So we're able to look at how different pathways are enriched in our cancer samples as well, and whether or not these could be potentially targeted pathways. So then finally, some of the study that we looked at in terms of all 5 patients, you can see clearly here, and we've just identified some of the hallmark chromosomal aberrations that you would see sometimes in uveal melanoma. So you can see clearly here that there is 2 patients who have chromosome monosomy 3, patient 2 and patient 4, and then we have chromosome amplification in chromosome 8 as well, and gain in patient 1, 2, and patient 4 as well. So then just to summarize of the patients that we have sequenced to date, and really there is publicly available data that we compare our own analysis to, and again we only have done this on 5 patients, and we have observed this normal trend whereby you would see either a mutation in the GNA11 or the GNAQ, but we'd just like to highlight 2 patients here that are a little bit more unusual. So our patient 3, basically we can see that this patient has neither a GNA11 or a GNAQ mutation. So maybe this patient, and we're going to look at this further, whether they have a PCL-beta4 or a CYSLT receptor 2 mutation. And although these are very rare in about 1 to 4% of uveal melanoma, they actually are usually mutually exclusive of GNAQ and GNA11. And then with patient 4 as well, we see that this patient has a loss of GNA11 and a mutation mutation in GNAQ. And again, that's quite rare because both these gene mutations tend to be mutually exclusive in uveal melanoma. So this information gives us a real indication of the different types of discovery and knowledge that we can utilize for the benefit of uveal melanoma patients. So newly developed medical treatments for disease, they do have limitations in terms of, changes between patients, difficulties in prediction of outcomes, and time-consuming drug tests. And organoid cultures really do have the possibility for utilizing, looking at a specific disease with genomic information. It really is a powerful tool for therapy, whereby we can now screen multiple drugs for responses to look at actionable targets in the patient's own genome. Thank you.